Okay, thank you all for joining us this week for the Miami Global Brain Tumor Symposium. Just have a short introduction here before we get going. My name is Michael Ivan. I'm one of the neurosurgeons here at the University of Miami and one of the brain tumor and skull-based surgeons. I'm also our co-director of our research program for brain tumors at Miami. I'm joined today by my co-directors, Dr. Morkos, who's professor and co-chairman of our neurosurgery department, also director of our skull-based and cerebrovascular program. Dr. Komatar, professor and program director of our residency, is also the director of surgical neuro-oncology. And Dr. Benjamin, who's assistant professor, skull-based surgeon, also director of our skull-based lab here called the Canes Lab at University of Miami. This is our 31st week since COVID started of doing the Miami Global Brain Tumor Symposium, and we couldn't do it without all the help of everybody, Christina, Roberto, Ingrid, Damari, and Ignacio. Thank you all so much, and the support from the Sylvester Comprehensive Cancer Center, University of Miami, and the Neurosurgery Department. For anybody who wants to learn more about our department, you can find us on our website. We're also very active on social media, Twitter, YouTube, Instagram, and the means. So please check us out there for more information about future symposiums. And that brings us to tomorrow. We have our cerebrovascular and skull-based symposium by Dr. Morkos. It's really going to be an outstanding group. He's been focusing in this last month of the year on complications, which I think is very appropriate. And he put together a fantastic panel to talk about complications during endoscopic endonasal surgery with a large range of experience there. And so be sure to tune in tomorrow night at 5 o'clock. This is actually going to be the last meeting for this year of the Miami Global Brain Tumor Symposium. We'll be returning monthly in 2021. So I hope everybody enjoys the holidays and New Year, and then looking forward to starting a brand new year next in 2021. For tonight, we try to make this as interactive as possible. Please use the Q&A button at the bottom of your Zoom. We'll try to get to all the questions that you may have. Once again, we don't offer CME, but you will get an email confirming your participation. And please be sure to like, follow, and share all our videos, which are on our YouTube channel, so we continue to grow and share all the outstanding knowledge of our speakers with everybody. So let's get to tonight. We have another outstanding group of panelists of young brain tumor neurosurgeons, starting with Eja Endom, who's coming to us from Emory. I need to update that. Sorry, Eja. He's now at Emory in leading their brain tumor efforts there as both in clinical and also in the laboratory. He just came from the NINDS and is bringing over a lot of experience from the NIH over to Emory. Dr. Zachariah, who's associate professor at Penn State and also co-director of their neuro-oncology program and director of their brain tumor and skull-based program. And Dr. Eichberg, who's one of our own senior residents here at University of Miami. He's on a 2-year NIH T32 fellowship doing research in my laboratory on gliomas. And he also just finished his clinical fellowship in brain tumors as well. So thank you to our panelists. Tonight, I'm very, very excited to introduce our speaker, Dr. Vogelbaum. He's a good friend of mine. He's also shares South Florida with me and leading the country in our brain tumor efforts, where he's in Tampa at the Moffitt Cancer Center. Dr. Vogelbaum is an internationally known brain tumor specialist. He's an investigator in laboratory research, translational, and clinical trials, which I think he's going to touch on tonight, specializing in gliomas, but all brain tumors. He completed his MD PhD at the University of Virginia in biomedical engineering, before then doing residency in WashU in St. Louis. He spent almost 2 decades at Cleveland Clinic, where he became really a world-renowned brain tumor specialist. While he was at Cleveland Clinic, he was associate director of the Brain Tumor Center and Neuro-Oncology Center and director of Center for Translational Therapeutics. He's currently, like I said, the chief of neurosurgery and program leader at the Department of Neurosurgery and Neuro-Oncology at Moffitt in Tampa, Florida. And he also leads on numerous scientific committees where we've shared a lot of acquaintances and work together. But most importantly, he's the co-director and co-chair of the Tumor Section Satellite Symposium coming up in 2021. I encourage you all to check that out. He's a principal investigator and co-investigator on a broad range of clinical trials, including investigator-initiated trials, most notably on blood-brain delivery devices. He's also inventor of multiple patents and medical devices for delivering therapy directly to the brain tumors, and he's co-founder and CMO of Infusion Therapeutics. He's published over 190 peer-reviewed papers, and his book is up there in the corner, principles and practice of neuro-oncology and multidisciplinary approach. He's also a recipient of several awards, including the American Association of Neurosurgeons Young Clinical Investigator Award. So very, very excited about tonight's talk because I think it applies not only to brain tumor specialists, but anybody involved in translational research, clinical trials. And so I thank you so much for taking out the time of this busy schedule to talk to us tonight.
Well, thank you so much, Michael, and thank you to you and your team, your colleagues at University of Miami, also to panelists, and really the opportunity to participate in this fantastic symposium, one that I've been following for some time. I think this was a great idea to put this together, and really happy to speak at it and hopefully can contribute. I hope you're seeing my screen okay.
Yeah, that looks great.
Great. So I'll go ahead and get started and talk about ways that neurosurgeons can engage in therapeutic development in the OR. And really, the reason for that is when we think about development of therapeutics for gliomas in particular, usually that's left up to the medical oncologists, and they're the experts in clinical trials, and you know, we pretty much help them when we can, but they really take the lead, and hopefully I can help everyone understand that we play a very important role in this. In fact, we can even play a leading role, not just a supporting role. So a couple of disclosures in terms of the usual sort of honoraria, but most importantly is the spin-off company that you mentioned, Michael, the Symposium Therapeutics, where I have indirect equity interest. Actually, it's owned by the Cleveland Clinic. But I do serve in a leading role for that. So I start all of my talks on gliomas with this slide, which gives sort of the overview of where we are today. And the truth is that for the past 20 years, about all I've changed on this slide is the date. Very much of it remains the same. And that is, it's still a dismal disease. The standard therapeutic modalities that we use are about the same as they were in the past. The only real recent change was the alternating electrical fields, the Optune. And in fact, in the recurrent setting, it's even worse. The only approved therapeutics in the past 2 decades do not extend survival. So really our most significant advances have been in slicing and dicing the tumor and understanding its biology more effectively. That is yet to translate into meaningful survival benefit. And many therapeutic approaches have been tried, including taking what works, like radiotherapy, and increasing the dose, or using brachytherapy or radiosurgery. None of those have shown survival advantage. Some of them have actually shown more injury than benefit. We've investigated a lot of biologics, including vaccines, other immunotherapies, many classes of drugs, none of which, unfortunately, have provided benefit yet. And so this is really where we stand in our field in terms of approved agents. And of course, in many other areas of cancer, I could never put all of this on one slide. In neuro-oncology, unfortunately, we can. And you can see that some of the approvals were based upon response rate data, but most of them are based upon survival. The bevacizumab was actually based upon durable response rate, but when the survival study was done, that one actually ended up showing no survival benefit. The only successful agents we've had have been cytotoxic chemotherapies and radiotherapy, which is a cytotoxic. Everything else has failed so far. And so I've thought about this for a long time in terms of what is the challenge? And when you look at it from a surgeon's perspective, I think that it becomes pretty apparent that the challenge is the infiltrating disease. It's the part that we don't really model effectively in our preclinical models. The bulk tumor, what is shown here, we have a very effective treatment for that, right? We take it out as surgeons. It's the part that we can't take out that ultimately is what leads to patient death and progression and then death. And that's what we really need to be treating. And most of our models really only look at this part, the solid tumor, the enhancing tumor, and don't really effectively look at infiltrating tumor when we're doing preclinical development. So So I think that's one of the most important factors limiting our progress, is that this is inherently an infiltrative disease. And so we do maximal safe surgery. It does provide survival benefit, but never a cure. And in fact, we think we know where the infiltrating disease is, but the truth, we really don't know all of it. We use T2 and FLAIR as a guide, but even the radiation oncologists know that's not enough. That's why they go 2 centimeters beyond that when they treat. Also, there's a lot of genomic and epigenomic heterogeneity in these tumors, which makes targeted agents very difficult. And this is actually a great example of how widespread these tumors can be. You can actually use molecular genetic tools to find isolated tumor cells. Obviously, this is an autopsy specimen, but you can find tumor cells well away from any place that you might have seen an imaging abnormality. So the implications of infiltration is that the complete resection of enhancing or non-enhancing, in the case of low-grade gliomas, tumor can never be thought of as a curative procedure. And in fact, there's been a lot of interest in doing supramaximal resection. That's the current debate that's going on. It still has not been convincingly shown to be curative. And it's not even clear whether the benefit is related to the procedure itself. So as I mentioned, one of the factors is infiltrative disease. We also know that we have poor access of therapeutics to the central nervous system. It's been estimated that more than 90% of potential neurotherapeutics cannot get into non-enhancing brain, and that's where the tumor infiltration is. And as I mentioned, our preclinical models of glioma fail to account for that issue. Also, our typical clinical trial design fails to account for the lack of therapeutic access. We don't spend enough time trying to figure out why a promising therapy that looks so great in the preclinical setting, and in which millions, if tens of millions of dollars have been invested, and then it fails. And we don't ask why. We don't really build that into our trials. And we've been doing this over and over again, and nothing has really changed. And so, so in a sense, our approach is a form of insanity. So one of the questions I really started to address years ago is whether our failed clinical trials are a failure to understand tumor biology or a failure to deliver. And so as part of this work, we started looking at this question, this claim that a new drug crosses the blood-brain barrier. It's made very often. We now use the term brain-penetrant drugs. All right, so let's look at this. You can go back in time to some early studies done actually this is the late 2000s, where in a high-impact journal, it was stated that in this clinical paper that post-treatment specimens were analyzed for the presence of intact drug in tissue. And in fact, it was detected in the tissue. And so this was considered evidence that the blood-brain barrier was crossed. Here are the data, and there are a few things to be aware of. Number one is these levels are pretty variable. Secondly, this was only from enhancing tumor, where the blood-brain barrier we know is defective. And thirdly, there was no attempt to look at the biology, what this actually did to the tumor tissue, and also there was no attempt to understand where was that threshold that you needed to have in terms of concentration. Was it here or was it here? And none of that was really looked at. So this was actually in a high-impact journal. In the preclinical setting, we have a lot more control over the environment, and we can look at this more carefully. So here is a study that was done in a preclinical study where this drug was claimed to efficiently cross the blood-brain barrier and was highly effective in xenografts. Okay, well, let's look at the data here. So here's the drug and the control versus the drug, and looking at tumor volume, and then looking at survival, and you can use all sorts of nice tools for that. But here's the actual data looking at the drug penetration. So in tumor, I would agree that there's a good amount of drug in that tumor, in that enhancing tumor. That's a tumor mass. When you look in the opposite hemisphere, there's none. That's normal brain. And then when you look in brain around tumor, if this were infiltrated brain, that's where your tumor infiltration would be. There's really no drug penetration there. So does it really cross the blood-brain barrier? No, it doesn't cross it at all. it gets into an area where there's a broken blood-brain barrier. So much of our terminology has been very misleading in our field, and that creates a problem. Here's what these models look like. This is a cross-section on MRI through in this case it's a rat, and here's the tumor that we implanted. And so when you do these kinds of studies, when you do a surgical biopsy of treated tissue, or you just take tumor out without perfusing with saline first, there's a confounder, and that is blood levels of drug. You don't know if you actually had drug that was in the tumor tissue. So we approached this initially when I was in Cleveland, running a series of studies. This was actually the drug. Most of you won't recognize this drug by this name, but you'll know it as erlotinib, which was one of the first EGFR inhibitors that made it to market. So we had this before it was approved, and we were studying this in our model. We had U87s overexpressing EGFR, and we gave the drug, and then we extracted the drug from the tumor tissue and from the brain. We did saline perfusions and so on, and we also did looked at the signaling effects. But I'm just going to show you the drug levels that we measured. So here's the drug in the tumor core. This is without perfusion. This is in the brain around tumor, and then this is in normal brain. And you can see there are two things. First of all, you see much more in enhancing tumor than you do in normal brain and tumor around brain. But then when you perfuse with saline to wash out the intravascular drug, this drops down significantly, substantially. Okay, so does this as well. So you actually were measuring a large component of this that was actually intravascular drug, not drug that was in the tissue itself. And here are the numbers here. Now, I'm not going to show the signaling effects. I can tell you that the dose that we used here had a moderate effect on EGFR phosphorylation, but it didn't completely eliminate it, and it did nothing to downstream signaling. We could completely abrogate EGFR phosphorylation with a higher dose of the drug. It also killed the rats. It was too toxic. This was all being done while there were clinical trials being done looking at this same drug in patients with GBM. There was one high impact paper in New England Journal of Medicine that purported, it was in New England Journal of Medicine that purported to show that there was a subgroup in which it was effective that ended up not being true. It wasn't in fact effective in anyone because that drug is very much a substrate for the pumps that are part of the blood-brain barrier, and it just gets pumped right back out. So looking at a drug level can be done, but it can be a little bit perilous. On the other hand, one can do pharmacodynamics, which is looking at the impact of therapy on treated tissue. And that's particularly the case actually for signal transduction inhibitors. That's a great thing to do. May also apply to conventional drugs, which are dirty, but you can look at assays for impact on DNA. And then of course, for immunotherapeutics, you can look at the tumor microenvironment. Outside of neuro-oncology, this is routine. You can't imagine a drug that's ever been developed in all of oncology outside of neuro-oncology where you did not carefully study pharmacokinetics and pharmacodynamics. It's just part of the deal. In neuro-oncology, it has not been the case, but it can be done. And this was some work that I did together with David Reardon, again, back this was published in 2012. We started this trial somewhere around 2010. This was actually a new mTOR inhibitor that was being developed. And so this was a phase 1 trial. We called it a perisurgical because we actually gave drug before surgery. And we have a new term that we use for that now, and I'll talk about that some more. And it had, the typical sort of phase 1 endpoints, but we did give treatment beforehand and looked at tumor tissue. And here's what we found. We compared to an archival specimen. The challenge we have in neuro-oncology is we can't take serial biopsies like you could with a melanoma or with a blood cancer where you just do a blood draw. It is harder, but it can be done. So we went to an archival specimen. We looked at one of the downstream signal transduction proteins in the mTOR pathway, S6, and we looked at its phosphorylation level. And in the archival, it was high, and in the surgical specimen, after we treated with this mTOR inhibitor, it was low. Unfortunately, for another downstream element, it didn't really change. Now that trial actually, that drug went nowhere. It had issues with the drug, but in the end, we really felt like this was not going to have promise for our patients in neuro-oncology. And as I mentioned, one can also do this in the setting of immunotherapeutics. There's a very busy slide. I'm just going to, it's really just to introduce the idea that I've been studying an element of the immune system called myeloid-derived suppressor cells for a number of years, and then partnered up with Jason Lafia, and we've been looking at this in relation to the tumor microenvironment and glioma stem cells. And based upon the work that we did, where we not only showed that MDSC levels are predictive of patient outcome and disease progression, but we could start to do detailed analyses of the immune system and identify potential mediators of this. In this case, we did a preclinical study where we were giving a low dose of a drug called 5-FU, which selectively depletes MDSCs. And when you give it at a low dose, it's not bone marrow toxic. This is a drug that is known not to cross the blood-brain barrier, so it has no direct anti-tumor effect. But we gave low dose to try to suppress MDSCs, and this is flow cytometry. Just take it for granted these are the MDSCs, and at baseline you have 18.4%. And with this low dose, it drops down to 3.86. You can suppress them further, but this is bone marrow toxic at 50. This is the standard clinical dose in the murine world. And so when you do this low dose like this, you can actually see at the low dose tumor suppression just by suppressing this element that suppresses the immune system. So by reactivating the immune system against the tumor, you actually get a survival advantage. Interestingly, when you go up to the full clinical dose, the animals die, and they die because of bone marrow failure, because that dose is toxic to the bone marrow. So we did this preclinically. We looked at the tumor immune environment and looked at CD8 cells in the tumor and how they went up, and Tregs went down. All looked great. We turned this into a clinical trial. So this is work that I did with Dave Peerboom, who's a medical oncologist, neuro-oncologist at Cleveland Clinic. And we did a very similar study in patients where we gave capecitabine, which is the oral form of 5-FU. We gave low dose before surgery. We collected tumor. We did blood collections all the way along and did post-op treatment as well. And this pretty much summarizes, this is now published work, but it summarizes the fact that with the MDSCs, we saw suppression with that preoperative dosing of the capecitabine. You can see it in more detail here. How at 300 milligrams, 450, and just to give you an idea, this is still about 20% of the dose of the clinical dose that would be used for colon cancer, and so we were able to demonstrate MDSC reduction, and then when we did more detailed analysis of the immune system using a technique called CyTOF, we were able to demonstrate that we had elevations in the effector components and reductions in the suppressor components. So this looked like it was activating the immune system against the tumor. And we're actually moving on from there, looking at a more specific way to really attack the relationship between glioma stem cells and the MDSCs. None of this is new, okay? These are not really new ideas of doing these window of opportunity trials. There's a term called phase 0, which we're moving away from for a variety of reasons. It's more of a window of opportunity clinical trials, and that is to give what is a therapeutic, thought to be a therapeutic dose for a short course of treatment. So it's not considered a cycle of treatment and not a prior therapy before a planned surgical procedure. Fred Lang wrote this very nice review in 2002, as you can see, describing several different ways to do these kinds of window of opportunity clinical trials. So I led a RANO group that asked the question, how are we doing? Okay, 18 years later, how are we doing? And really this all just talks about the rationale for doing this. This is the group that was involved in doing this review. We just published this this year. And what we wanted to do was look back and say, okay, how often did we really do this in the setting of drug development? So over 25 plus years of analysis, only 22 drug trials included tissue-based analysis of drug levels or biological impact, less than 1 per year. And that included analyses in blood, CSF, tumor tissue, or tumor-infiltrated brain. Now, because the blood-brain barrier really limits penetration, you really want to make sure you're at a full therapeutic dose when you do this, but only 50% of the trials use the full clinical dose. Tissue samples from non-enhancing tumor, truly behind an intact blood-brain barrier, were analyzed in only about a quarter of these trials. Drug levels were assessed in 75%, but biological effects in only 68%. And there were just notable missing pieces to the analysis of almost all of them. And none of the claims about likely efficacy based upon limited data held up in phase 2 or phase 3 studies. So the truth is, as much as that concept has been out there, we've done a very poor job in our field of using that approach. And really what it comes back to is this infiltrating tumor problem. Many of those trials, they were sampling only enhancing tumor, okay? But we know we can take that out, and this is the part that we need to be sampling, is the part where the drug really needs to get to. And this is becoming more and more important. It's not the only issue. We also know that we have a problem of heterogeneity. We can see this demonstrated at a genomic level. We can even see it at a radiomic level. This is some work from colleagues here at Moffitt doing radiomic analyses of these tumors. And you can have areas that are outside of the tumor mass or parts of the tumor mass that are biologically better or worse than other parts. And so we need to do a better idea of understanding what's going on in those areas biologically and therapeutically. This is an example from UCSF showing how they could take biopsies from different areas within the same tumor and see differences in the genomics and other markers of the tumor activity. So what this really points to is we need to have a better way of classifying our tumor samples when we're working in the OR to do these kinds of window of opportunity studies, but I think also in general for our basic discovery research. So one of the things that we started here about 2 years ago is this intraoperative database for multimodality data capture, what I like to call geotagging because it's just easier to say. And that is we now, with every case that we take to the OR, we actually collect information from the case, things that normally weren't collected in a database. We collect the information, including whether or not we used 5-ALA or something like that. We then will classify by location, and we have each sample that we take has a letter code to it, and this comes back. This can be referred back into the database. But most importantly, we have a screenshot. We have a screenshot from where we took that sample. So we are able to then go back with radiomics or other approaches to sort out, are we looking at enhancing tumor? Are we looking at non-enhancing, tumor-infiltrated brain? What is the radiomics appearance there? What else might have been done there in terms of radiation, for example? We have all sorts of questions we can ask now that we have a geotagged database of pathology samples. And so that's something we've been building up, and we're well over 100 cases now with that. This really gives us a tool to really start developing more robust window of opportunity clinical trials. We have a couple of these open right now. One of them is with a BET inhibitor from Celgene, and the idea is it blocks MYC activity, may also downregulate MGMT. So we are doing all sorts of analyses after we give the drug before surgery, and then go ahead and take out the tumor. And then the second one I'm going to talk about in more detail is a trial of this first-in-human antibody made by OncoSynergy, an antibody against the integrin receptor master unit. And this is actually, I'll show you the form first of what we do, where we're doing preoperative assessment and then treat, and then resect so we can see what happened with the treatment, and then treat again. We'll talk in more detail about that. Also, I want to highlight the fact that Michael Liven and I are part of this project to become part of this new U19 glioblastoma therapeutics network, if we're so lucky. The Moffitt and University of Miami teams put together a proposal for this. This is going to be a new NCI-funded network that is charged with performing early stage development of what are likely to be brain penetrant drugs that have not been used previously in glioblastoma. And the way that we interpreted this program announcement was that it meant that every initial clinical assessment of these new drugs had to include a window of opportunity design element. And so this required 2 projects. In both of our projects, that's what we're looking at, and then have a lot of really interesting and comprehensive omics analyses to be done on those samples that have been treated, and we can compare to prior material from those patients to see what the drug has done. So I think this is the way forward, and again, this is where neurosurgeons really have to be part of it. Now, as I mentioned, we're both asking the question of whether or not these drugs are actually have a chance getting to the tumor tissue, but then we also have to recognize the fact that many of them really don't when we give them systemically. And we need to look at other ways of trying to get these drugs to their targets in the brain. And so one of the other questions I've asked is, how can we as neurosurgeons improve therapeutic access to the brain? Well, there are a couple of ways in general that one can improve delivery to the brain. One is by manipulating the drug. I am not a medicinal chemist. I have no ability to do that. I know the history of that has been quite challenging. Very often, the manipulations you make to the drug to make them more brain penetrant actually remove the activity. There are some new drugs that are being developed that truly are brain penetrant. Some of them have some side effects in the brain that are a little worrisome. Some of them may not, and we'll have to see how that goes. But that's certainly an area of active investigation. There was some interest in using the drug and protein transport mechanisms to piggyback into the brain. That has not really worked out so well. Blood-brain barrier disruption, I was involved in that early in my career. It was a little bit messy, I would say, and didn't really work well for gliomas. It worked very well for CNS lymphoma. There are some other approaches being used now using focused ultrasound or LIT. It's still a little unclear how much access you really get with those approaches. That is work that needs to be done in more detail. And then there's this area of direct brain delivery. And the concept of direct brain delivery, putting the drug directly in some sort of release mechanism, was actually validated by Gliadel, the carmustine wafers. Of course, that predated the development of temozolomide, and the effect was modest, and they have overlapping activities, so temozolomide has become the standard. The idea of doing intrathecal or intraventricular injection has been problematic because the CSF is not really in homeostasis with the interstitial space in a way that gives kinetics that are likely reasonable for the therapeutics. And then there's this idea of convection enhanced delivery. CED was developed initially by Ed Oldfield at NINDS. The idea was that you would pump a drug of interest slowly into the brain. Now, that seems kind of obvious today and the like, but back then, and when this was conceived of, most of the neurosurgery was worried about how to get fluid out of the brain, because that was a bad thing. And so the idea of actually pumping fluid into the brain was completely counterintuitive, but it actually worked, because if you go slowly, you don't actually increase the intracranial pressure, and you actually get better distribution, more uniform distribution than just with diffusion. And so this schematic kind of illustrates the options that we have for introducing drugs directly in the brain. One is to do an injection. And when you do that, the brain is viscoelastic. It kind of resists injection. Most of it comes up around the needle. We don't really show it here. Most of it goes up this way. This, of course, is a resection cavity. These are the tumor cells that are in the infiltrative margin. you're not going to get much coverage that way. And there have been some recent clinical trials that have used that approach with replicating viruses and haven't really worked out. There was the wafer approach, which I mentioned earlier, which did provide proof of principle evidence. But again, it's a diffusion-based mechanism. You're not going to get much penetration of the drug of interest. And then the idea is convection-enhanced delivery. Now, it's easy for me to make a schematic and show this and say, hey, see, it works. You can do that with anything. I'll show you data later on that I think are going to be more convincing. So what can be delivered? Almost anything, conventional chemotherapeutics, but they must not cross the blood-brain barrier easily, which is good because there aren't many that do. The problem is actually now the reverse. You want to keep them in the brain. So you want things that don't cross easily. And most conventional drugs don't cross easily. The small molecule drugs could be, but on the other hand, if there are substrates for the active pumps that are part of the blood-brain barrier, they'll just get pumped right back out. I like the idea of nanoparticle formulations. I actually saw a patient earlier today who was asking me all about that. I think the idea of trying to introduce drug depots in the brain would be a great idea. It's just there are challenges in the formulation and the approval process. Oncolytic viruses, vectors, all sorts of things can be delivered this way. There have been a number of clinical trials that have looked at using CED, and these are the different classes of drugs. Many of them were created specifically for this approach. Some of them are old chemotherapies that have been used, and you can see the different phases of trials. There's only one that made it to completed phase 3. That's this one here, the syntritic and vasudotox. This one was actually aborted, but this one actually made it, and it ended up being a negative trial. And there were a number of factors that led to this. I was part of a failure analysis that really tried to look at this question. And there were a couple of important principles that we decided were very important, obviously, which was that we really couldn't do pharmacokinetics. We were just pumping a drug in with a tube that we pulled off the shelf, and we had no idea where it was really going. At that time, we could not co-infuse a tracer easily. And we had questions about the technology since it wasn't really, we knew that backflow was an issue. And here's that example I wanted to show where you can see when CED works, it really works. So this was, so John Sampson was able to co-infuse radiolabeled albumin in that trial with some of the patients. And here you can see a catheter, and this is after 48 hours infusion, beautiful infusion here, nice distribution. Here's the resection cavity. And so you can see there are a couple of problems. Number one is this area is not getting treated at all. This area is not getting treated at all. This area is not getting treated at all from that one catheter. But it showed that, this can work. Unfortunately, most catheters look more like this, where you had some initial infusion, but then the rest backflowed and was just over the surface of the brain and got nowhere. And the problem was we were just using tubes off the shelf, and we knew that those tubes were not really up to the task, but we didn't have enough evidence to prove that. Once we generated this, it became less interesting to use a tube off the shelf. And that was an opportunity to try to develop better technology. But the other thing was we had to be able to see where the drug was going. There were a number of technical and regulatory hurdles that made that nearly impossible. As is usually the case with progress, it was a patient with a connection to a U.S. senator that ended up breaking that logjam, and the FDA suddenly was allowing a tracer to be co-infused. There had been lots of preclinical work showing it was safe. So it ended up, Russ Lonser was able to do that and show that you could safely use it. Also, as I said, we knew that the off-the-shelf catheters were not really up to the task. This is work that, again, had been done early in the development of CED, showing that the size of the tube really mattered, actually the end port. And that was where the idea of a step-down catheter came from, having something very small. It does have its own issues, plugging during insertion and then introduction of air during stylet removal for a non-rigid catheter. Those are all problems, but at least the principle was there that we needed to go small. And so there were a couple of new devices that were developed based upon that principle. The step-down was first developed by MRI Interventions called Smart Flow Cannula. There's a second version of that that's more flexible, that's made by BrainLab. There's a MEMS cannula made by Alcyon, a chronic one made by Renishaw, and then I'm going to talk more about the one that we developed when I was in Cleveland. Some of these have 510K or CE mark clearances, none for its use as CED devices. We don't need to go into details of why that is. Also, it's important to know that this one here, the SmartFlow and the MEMS cannula, are rigid devices, can only be used in the OR, which means very small volumes of infusion, whereas the Brain Lab and the Cleveland Multiport can be used over a period of days. This one has only been used in the UK to date and is really a custom-made system for each case. It's not yet available commercially in the US. So I've actually had the opportunity to use the 4 that are commercially available. This is a case that I did in intraoperative MRI with the MRI interventions device. This was during the Tokagen trial, and we actually did CED of that virus. This is actually work from Mark Suedane, who's been developing a theragnostic for treating DIPG, and he's been using the Brain Lab version of the MRI interventions cannula, and really exciting work that he's been doing there. But ultimately, when we start thinking about glioblastoma, we have to think about the volume issue. Okay, so when we're treating, we're not just treating a rim here. We're not just treating this enhancing tumor. We're treating this whole area. This is where we need to get drug to. And so if you think about that, you have to think about what is that T2 flare volume. That's just a start. It goes beyond that. What is that volume? So that median volume, we looked at this in a series of patients that we were evaluating volumetrically with GBM. It's about 37 cc's median, but it's as high as 170 cc's. So you need to really be able to cover a very large amount of brain. And when you have a single source device that can be used in the OR only, and an infusion rate at really is the maximum is about 10 microliters per minute, the most you're going to get over 6 hours is 3.5 cc's. And so if you think of maybe a 2-to-1 ratio for that, maybe 3-to-1, you're not talking about these kinds of volumes. So we need something that we can, where we have time for delivery. So we set out to develop this device to achieve a reliable high volume drug delivery, not just for intraoperative use, that's backflow resistant, biocompatible, drug compatible, and can be used with conventional neurosurgical equipment, not just intraoperative MRI. This is a device we came up with. This was when I was at the Cleveland Clinic. We partnered with a global manufacturer, Parker Hannifin, and we formed this spin-out company called Infusion to develop and has multiple patents and most importantly, a 510K clearance. But making a device was not enough. I wanted to prove that it actually works and really understand how best to use it. So we put together this pilot trial strategy. We had to pick a drug. After all, there is no drug that's approved for direct delivery into brain tissue. And so we picked topotecan. This is based on Jeff Bruce's phase 1 work that he had done. It made it much easier. The CEDAR, the FDA liked that. We used gadolinium as our tracer, our device. We actually had some funding to do this. Managed to deal with all the conflict of interest issues. The biggest challenge was to get the device people and the drug people at FDA to talk to each other. They wouldn't talk to each other. We actually had to force the issue. They finally did. And we got the trial up and going. And this first pilot study we published recently in Journal of Neurosurgery, just where we put one catheter into enhancing tumor, one into tumor-infiltrated brain, same infusion rates, and we saw complete differences in extent of volume of distribution as we expected. We then went on and did a second protocol where we took out that enhancing tumor, since we know that's an effective treatment, and then put 2 of the catheters into the tumor-infiltrated brain, and now started getting volumes of distribution that were unprecedented. And that was a lot of fun to be able to do that. Now what we're doing is we're going back to the enhancing tumor, and we're infusing at a much higher rate, and we find that we can overcome the leakiness of the blood-brain barrier and actually get distribution not only in the enhancing tumor, but also the tumor-infiltrated brain. This is one catheter that was used in this case. We've done 2 of them now, and we're looking to go beyond that. All right, so now let's talk about therapeutic development. This is really the exciting part. So now we have a device we know that works, and really we need to start thinking about maybe better drugs. Topotecan, I think, has some positives. It also has some potential negatives. Let's try to really target the biology. And so we're working together with a company called OncoSynergy. It's a biotech startup that's led by Anne-Marie Carbonell. Anne-Marie was actually a neurosurgery resident with us in Cleveland, who ended up via a few steps becoming a leader in biotech development. And so she and I put this trial together. When I say she and I mean she. Really, took their lead compound, which is OS2966. It's an antibody that targets the master integrin subunit and involves a number of interesting design features to the trial that we both were excited about. One is direct delivery. Second is the window of opportunity design to really look at what's going on. And so this is actually a slide that she provided where we're initially doing this first-in-human study with the usual sort of objectives that you would have with a first-in-human study, but also with the pharmacodynamics. These are recurrent GBM patients, the usual criteria. We're going to start by doing a biopsy, and so we'll have pre-treated tissue. We'll then do an infusion into the enhancing tumor, cover that, as we showed we can do now. And then we'll come back a day or two later and resect the tumor. And now we'll have a treated sample to look at. And then we'll go ahead and treat the tumor-infiltrated brain as well and have typical clinical endpoints. Again, this is a timeline showing that in more detail. And this is just some examples from our initial studies when we were validating the device. We've just opened this trial, the OncoSynergy one, and hoping that we'll have our first accrual shortly. And as part of that analysis, Anne-Marie and her team have a detailed understanding of what kind of markers to look at when they use this drug. They've done a lot of preclinical data and evaluations of this, and so they'll be able to look at the ability of the drug to bind to its receptor, and then look at downstream signaling, and then, of course, ultimately functional assays as well. And then we'll have the usual clinical outputs. So endpoints, safety, feasibility, PFS, OS, and then the biological as well. We're starting this here at Moffitt Cancer Center, and I did not talk about some of the accelerated titration design to this, how we're going to go up pretty quickly in the doses initially until we find what is likely to be a biologically effective dose. And then at some point, we're going to look to expand to other centers after this initial experience. We're currently screening patients, and I have a couple of numbers here and ways to reach me. And so if you know of anybody, I would love to talk to you about them because we're very eager to get our first patients on for this. So in summary, I think, as I said earlier, our field is plagued by failure to learn from failure. We as neurosurgeons have the opportunity to really change that by doing these kinds of window of opportunity trials to understand the biological effects of novel treatments and whether or not they really have a chance of working. And then we also can develop therapeutics that can be delivered directly into tumor and tumor-infiltrated brain. I should add one of the points in terms of the window of opportunity. There are ways to do that without a surgical resection. I think Edja is going to talk about that a bit as well. So there are ways we can do it both over a period of time and also in the OR. But again, neurosurgeons are key to that. So I'd like to make a few acknowledgments. This is the team I work with here at Moffitt, which is very multidisciplinary, fantastic. We're really excited about growing our program. We have medicinal chemists here. I've never had an opportunity to collaborate with one before in this way, and so we're developing some drugs that can both be delivered by CED and also ones that might be able to be given systemically and have a chance of getting in the brain. A number of the funders are listed there, and hopefully, if you haven't figured out the license plate yet, we can stop cancer in your head. That's our ultimate goal. And I'll leave you with a few shots from Tampa. And this is like every night. It's like Disney. We have a parade every night, and then we go to the beach and watch the sunset. So with that, I'll thank you for the opportunity to participate.
Okay, yeah, like
every
talk I give, I always say, you'd think I'd have Zoom figured out now in month, whatever it is of the pandemic, but we're still working on it. So I'm going to give a couple thoughts on clinical trial development for neurosurgeons and give an example of a trial that I'm running that Dr. Vogelbaum alluded to. I like to say it's one of my big soapboxes that it's really critically important that we understand what our drugs are doing in tissue. And I often tell residents that a lot of neurosurgery is actually a religion. It just kind of depends on what temple you've trained at. And hearing Dr. Vogelbaum's speech, it's like it's like going to the temple. It's like this is someone who's been studying this and been on that soapbox and telling people that this is what we need to do for a long time. And so it's good to see another wonderful presentation of all the work that's been done over decades in this area. So really quickly. I am going to try and show you a little bit about some of my thoughts on one of the ways that we can look at how we're actually doing in tissue, kind of in a pharmacodynamic way. And that's with microdialysis. Similar to what Dr. Vogelbaum talked about, convection-enhanced delivery, this is not a new technique. This is something that's been done for a very long time, actually mostly in the trauma literature, where they've used catheters that have semi-permeable membranes, and then you slowly perfuse something into it that's about isotonic to interstitial fluid, and then slowly allow that to come out. And in doing so, you allow solids to very slowly diffuse across the membrane, so that you get an idea of what the concentration of your target analyte is. So the safety of this has been well established. I actually participated in a clinical trial at the NIH already. We're looking at temozolomide concentrations after the delivery of a blood-brain barrier opening agent in recurrent glioblastoma patients. And it's been done for cytokines as well. So cytokines, as I'm sure many or all of you know, are one of the ways that our immune system communicates with each other and either stimulates or shuts down an immune response. And in the trauma literature, there are a lot of consequences from neuroinflammation. And so there have been several studies that have tried to look at what the cytokine changes are in brain tissue over time after traumatic brain injury. And this is one of the studies from about 15 years ago. This has also been done in brain tumor patients. This is a study that someone did fractionated radiation to metastasis patients, and they were able to see that there were different responses in cytokines to patients that had microdialysis catheters in their tumor and also in the brain around the tumor. As Dr. Vogelbaum stated, it's really important that we know what's going on in the brain around our lesions as well. So I'm interested in immune therapy. Immune therapy is obviously very big right now. Checkpoint inhibition having won the Nobel Prize, I guess a year ago now. And so we wanted to figure out checkpoint inhibitors, they're being used in glioblastoma, there's several different combinations and studies. Are these actually having an effect in brain tumors? And after looking at the preclinical models and seeing that interferon gamma rises were one of the things that denoted a successful response to checkpoint inhibition, we wanted to see if we could measure interferon gamma in brain tissue. So what we do is similar to Dr. Vogelbaum's study that he's just getting up and running now. We take a biopsy from the brain tumor on a patient who we think has a recurrence. I say think has a recurrence because oftentimes we think a patient has a recurrence and they actually don't. We actually have had 5 patients who were recommended to our study by our tumor board and the neuro-oncologist, the neuroradiologist, and myself. We all agreed, well, this is someone's tumor that's going to need to come out surgically, and biopsied those patients in several different locations, necrotic core, on the border, contrast enhancement in multiple different locations, and found that they really had treatment effect. And in following those patients over time, those 5 patients, those areas that we sampled did not blossom into full tumor. And this was not a sampling error. They truly only had treatment effect. And there are separate patients that actually have real tumor. So I think Dr. Vogelbaum is up to something or onto something when you talk about having to sample a lesion at the upfront part of the trial at the time of recurrence before you go on to treat them. So at the time we do the biopsy, we place a microdialysis catheter, one into the contrast enhancing tumor and one also in. I'm going to take that contrast enhancing tumor out. So I have to know if the drug is having an inflammatory response on the brain around the tumor as well. We then give 1 dose of nivolumab, and then 5 days later, we take the tumor out. And this is just another look at the study scheme. We do a biopsy day 1. We place a lumbar drain, so we can also look at CSF and cytokine changes in the cerebrospinal fluid. We take a blood baseline. This study I came up with and was in part initiated by Dr. John Lyons, who's actually going to be a fellow with Dr. Vogelbaum next year. I'm very excited for him to have seen the way that we do our surgical trials and then to learn from Dr. Vogelbaum so that I can then learn from John in the future. But again, we have tissue before. We give that immune therapy in the middle of the trial on day 3 after we've let the inflammatory response of placing the microdialysis catheters go away. And then we take the tumor out day 8, and then we continue to follow them with nivolumab and anti-LAG-3 from then on. Obviously, there's several potential outcomes, and really, no matter what we end up seeing, whether it's some patients have an interferon gamma response and others do not, we can follow those 2 groups clinically. Again, this is a pilot trial. It is obviously not powered to show us, say, the interferon gamma responding tumors do better than the others. This is mostly for safety and efficacy, or just the feasibility of measuring this. All the patients could show an interferon gamma response, which would be interesting because that would mean the drug is having an effect in every patient, but maybe clinically it's not showing up. Or no patients show an interferon gamma response, and that might just be that maybe immune therapy is the way to go, but systemic delivery is just not going to do it for brain tumor patients, and we may be looking at catheters for immune therapeutics as well. So just going back really quickly to I think the beginning of Dr. Vogelbaum's talk, I think it's really important that neurosurgeons are involved in clinical trials. You see a lot of these clinical trial working groups and things that are done by neuro-oncology, and it's almost all neuro-oncologists and Dr. Vogelbaum, and maybe Dr. Sinai, and that's about it. And I think there should be more of us in those rooms because it's really only neurosurgeons that can talk about the certain questions that we can answer that can't be answered without us, and certainly kind of makes you attractive for a job hire in the future. And then just sticking on the same thing, whether it's sample collection or drug delivery, I think it's really important to consider your advantages of being a neurosurgeon when you're thinking about moving into clinical trials so that you can differentiate yourself. And I'll stop there in the interest of time and let us get to hopefully questions from the rest of the panelists and the attendees. So thank you for allowing me to give my little nibble of my entree into this field that Dr. Vogelbaum has done such wonderful work in for quite some time. I'm looking forward to a new era of continuing to do these trials in the future.
Thanks, Edja. That's really great. I mean, I completely agree. There are certain questions that come up on the day-to-day treatment that having had a surgical perspective, having been in there, correlating the imaging with the clinical picture, that really the neurosurgeons are able to get a better sense of what the true questions should be and how the trials can be modeled accordingly. Dr. Vogelbaum, I had a question for you, and feel free, the panelists, everybody who's listening, if you have any questions, please type them out in the Q&A box and we'll be sure to get to them at some point. But in your database where you were keeping track of the different pieces that were sent to the pathology, did you guys, on analyzing that data, did you notice like any patterns, or did you notice anything from the radiomics come out of that? I mean, I think that's really interesting. We tend to do that, think, oh, okay, there's an enhancing nodule here, we'll take this as a separate specimen. And then again, we don't go back and necessarily analyze that and see where we right, where we wrong, and make, try to sort of make a conclusion about these different specimens.
That's a great question, and that's exactly what we intend to do. Right now, we're in the collection phase, and Edja, I agree with everything you pointed out, and yes, that could be a great project for John when he comes. We should have built it up enough for him to start addressing that, because that's exactly what we want to do. There are all sorts of basic questions that we haven't really looked at yet in detail. How different are the samples from each other? We do see them, actually our pathologist, Rob McCauley, often when he's presenting the case at our tumor board, he'll have a PowerPoint with all of the different samples there, and you can look at them, and morphologically they look different. So we could start asking more detailed questions about how different are they really, and how do they impact on the tumor biology and correlate with the radiomics evaluations and so on. And this is also going to play into another effort I'll plug right now, which is another RANO effort to try to help define what a recurrence means pathologically. Because as we all know, when we send that sample intraoperatively in a quote unquote recurrent high-grade glioma case and ask the pathologist, can you tell us, is there a tumor here or is it treatment effect? They always struggle at that, right? And I don't blame them. It is a struggle. So we're trying to come to some consensus about what is it that really needs to be evaluated, and how do you correlate the findings on pathology with outcome. And there are a number of pathologists who've been really trying to address that question. So we're now working together to try to figure out how to address that.
Yeah, that's very exciting. All right.
Yeah.
Dan, do you want to go ahead and present your?
Sure, absolutely. Let me just share my screen. I have a few slides I wanted to show to sort of give us background. Can you guys hear me?
Yes. Yeah.
Okay, perfect. So
your screen, your slides though aren't up yet.
They're not up.
There you go. Perfect.
You can see them?
Yeah.
Mm-hmm. Just go to presentation
perfect.
mode now.
Perfect. So I guess my question for Dr. Vogelbaum has to do with GVM heterogeneity, and that's one of the things that we're looking at in the lab with Dr. Ivan. So we're looking at a protein called CD97, which is something that Dr. Ivan was looking at with Dr. Parsa when he was at UCSF, and they looked at the TCGA database and they found that higher CD97 expression was associated with a worse survival rates. Then they also found that in GBM, like U87 cell lines, higher CD97 conferred a higher in vitro assay invasion rates. So we decided to get some patient-derived glioma stem cells and sort of look at CD97 in this model. U87 cells are because they've been cultured for so long, relatively different genetically from the original tumor that they came from. So we wanted to look at freshly obtained patient-derived stem cells, and we found that in the patients that had the cells harvested, the ones that had more aggressive radiographic invasion patterns tended to have higher levels of CD97. And in an invasion assay that we did, the invasion rates also correlated with the levels of CD97. And then we were able to manipulate using shRNA the levels of CD97 to knock down those levels. Then we used a flag tag protein with a CMV promoter to overexpress the CD97 in an mesenchymal subtype patient-derived glioma stem cells. This correlated positively with impact on invasion. We also were, looking at EGFR and GFAP and SOX2, and these levels also appear to correlate as well with CD97. I guess the question that I wanted to ask you is, clearly there's a failure of drug delivery, which you and your team have done a very fascinating endeavor to help fix. But how much of the problem do you think is also heterogeneity? So, for example, a lot of the therapies that we currently have target rapidly proliferating cells. Radiotherapy, temozolomide, a lot of the standard chemotherapies target rapidly proliferating cells. But what if the invading, migrating cells are not proliferating as rapidly? What if they, in other cancers, there's sort of a epithelial to mesenchymal transition where cells kind of switch from a more proliferative phase to a more invasive phase. So I guess I'm wondering what future endeavors might help to target GBM heterogeneity.
Yeah, I think that's an incredibly important point, and that you're touching on. And one of the comments I generally make in the talk that I give, or parts of it, is the fact that the only drugs and therapies that have worked to date are the non-specific ones, the DNA damaging ones, the cytotoxic ones. whether it be radiotherapy, tumor treating fields, or chemotherapy, cytotoxic chemotherapies. Our targeted drugs don't really work, and it's a little bit of code for I'm skeptical about targeting one drug. Now, targeting one target. Now, we may be able to do that. There may be other aspects or secondary effects of targeting a specific element that may be very important here. So I think there's still a lot to be learned. We have to look at the treated tumor tissue to figure that out. Is the single drug alone going to be enough, or is that going to, say, predispose to some other kind of stimulus to take effect? I mean, that's the idea of altering the immune microenvironment and then adding an immune therapy. I mean, those are the kinds of questions. So, but the idea that we can give a small molecule drug and we're going to cure a tumor there is potential with the H3K27M tumors, for example. There's been some really remarkable responses to that, but that is a tiny fraction. It's a very different biology, I think, from the rest of them. Arnie Atami here at Moffitt is a physician scientist who has been looking at this question of mesenchymal proneural transition or proneural to mesenchymal transition. Has some really interesting mechanisms that he's been studying that control that transition, and it seems to be very important in GBM. So I agree. I mean, anything that we do to try to understand the impact of heterogeneity is going to be very important ultimately in terms of what kind of targets we choose.
Thank you.
Yeah, that was a fantastic talk, and I think hits on a lot of the reasons why it's so important for neurosurgeons to be doing this. I appreciate that. I would also add, somebody who, anybody who does a lot of clinical volume of brain tumors, you kind of need this because how unfortunately ineffective kind of the things we do are. And so, like you said, you just are kind of, kind of going crazy if you don't feel like you're doing something else to push things forward. I just have a couple more philosophical questions, some things to kind of follow up on what Dan had said. So, along those lines, with most trials, you're forced in many ways to do single agent trials. With many cancers, patients are getting multiple treatments with systemic cancers, non-CNS malignancies. I mean, it is the norm that you require multiple agents, that not a single agent is going to be effective. And so why should we think it different in the brain? And I know there's obviously difficulties in designing trials and getting multiple agents approved, and we didn't get into any of the regulatory stuff, but it becomes even more complex. What are your thoughts on that? I know there's not an answer, but just curious on your thoughts.
Well, I'll give you there are two aspects to that. So it's a very important point. You're absolutely right. Not only do we need to understand what signal drugs do, but also combinations. So there is a trial that Justin Lathy and I are putting together along with some collaborators here at Moffitt, Solmaz Sahebjem and Filippo Veglia, looking at a combination of an agent that targets signaling between GBM stem cells and MDSCs, and an immunostimulatory, a PD-1, essentially a checkpoint inhibitor. We're doing the same kind of design for this, which is going to be doing pre-surgery treatment, but now we're going to randomize that pre-surgery treatment to either individual agent or combination. And then the post-surgery is going to be combination, right? Yeah. So the design of that is very straightforward and could be really interesting because now we're doing that analysis that we always do in the laboratory, with mice. Now we're doing that in a way that hopefully is going to be very informative from patient samples. And then they're all getting treatment from that combination, which we think is effective, or hopefully it will be effective. I'll tell you what the other challenge is, though. So designing it is easy. We now are having to deal with two different drug companies. That's hard.
Yeah.
And that's ultimately one of the problems that we have. I saw that when I was doing that early translational work where I showed the the rat xenograft model, I was doing individual drugs and seeing things, and then of course their biology was all related. The signaling was all related, so I started doing combinations. One of the companies found out I was doing that and went nuts. They actually were threatening lawsuits and all that. That was my, my naive approach, not understanding that there was a lot of IP involved, and these were drugs that were not yet approved, and they went nuts about it. And so we were not allowed to do that anymore in those preclinical efforts unless we had some very complex multi-way MTA in place, which never could happen, right? In the clinical trial setting, there may be a little bit more flexibility, especially with the types of drugs that we're talking about, because a lot of the IP is already locked down. So we're hoping to be able to pull that together.
Interesting. And the other question is kind of, again, along similar lines of the kind of some of the complexities that I think we're forced into. Do you feel that with the nature of many of these trials, we're doing them on recurrent patients, and that maybe represents a separate group of patients, and that maybe we should be doing many of these trials on patients up in the upfront setting before we've self-selected for maybe a more inherently treatment-resistant cohort of cells. And so I know that's just the nature of kind of how trials tend to be done historically. We start with the recurrence and the patients with the least options, and then we work our way backwards. But what are your thoughts on doing these more upfront?
So Brad, you might have been in the OR when we had the Snow Town Hall on this topic of clinical trial design. And that was very much a focus of that session. All of the issues relating to the fact that we have therapeutics that have failed in the recurrent setting that came up front and have shown more success. We have lots of reason to believe that biology is different and more complex, of course, and it's now drug-resistant tumor, right? Because there was a drug that was working and now it's resistant. So it's different. Lots of reasons to do that. There are challenges to bring it up front as well, though, because now you can't really take away the standard therapy. So now you have to understand whether or not your new therapy works well with the chemo radiation. You can make the case for MGMT unmethylated patients with just the radiation. That can be done, but you're still doing an interaction. Okay, you can't just do that alone. And it's also a little bit more complicated to do the window of opportunity because the resection's already done, and now you're asking about doing this treatment. Although there are ways to get around that because we think that ultimately the window of opportunity is asking much more fundamental questions like, does it even get there? And is it having any biological effect? And so those could probably still be done in the recurrent setting, whereas we bring the development up front. And so I don't know if any of you are going to be on the study section for the U19. I don't want to give away too much, but that's kind of the design that we did with the Moffitt team and the University of Miami team, was to say we're going to do our efficacy-oriented clinical trials in the newly diagnosed setting and our biological evaluations in the recurrent setting, just to verify the drug gets there and understand what it's doing when it gets there.
Yeah. I'm going to ask one very boring question in many ways, but I think very relevant to what we're talking about. For trials like that, so let's say newly diagnosed, I'm out in the real world for the first time. I was at the NIH for a while, and no one asked me what insurance paid for, didn't pay for. But so, scenario where you have a newly diagnosed patient who doesn't have mass effect, you radiographically diagnosed their tumor. Is there a way to biopsy that patient before you put them on a window of opportunity trial in the newly diagnosed setting to see if you get a signal, and then resect them, and then treat them with standard, but maybe you have something that you have a signal that you can use at recurrence, or maybe impetus to use that in a new trial up front in the future? Is there any way to get that paid for?
I don't want to go too far into the weeds of that, because there not only are payment issues, but in my mind there are some ethical issues as well. But in general, there was a time when we would biopsy these cases and then take them to surgery. That practice is pretty much gone. That's sort of like the idea of being able to admit the patient to the hospital the night before. Many of you probably didn't even do that in your training. I did. I do those H&Ps the day before, right? That's long gone. And the same idea with the biopsy before resection is so it would be challenging, and then figuring out how to get that paid for. And I'm not sure in the newly diagnosed setting that's really in the patient's best interest.
Sorry, I missed some of that. We have a couple more questions here. I don't know, Carolina, did you get to
Yeah, so we have not gotten to the questions yet, Mike, but everybody's sort of given their spiel.
So Nalia asked, who's also collaborating with everything that we're doing, what are the regulatory approval hurdles for getting 2 drugs approved to be administered versus, via the methods and devices that you mentioned?
Yeah, so the way the FDA looks at it is when you go to your registration trial, that is typically a phase 3, it might be a phase 2 in some cases, but typically a phase 3, when you go to your registration trial, which is the trial you do to get approval, whatever you're doing in that trial, that's the label, okay? So if for example, and I've been to the FDA multiple times with my device, but also we set up a drug company. We went to them as a drug company as well, because I wanted to see how they would approach it from the drug side, right? And we asked questions like that. So if we had a drug and we were co-administering gadolinium, okay, and we did our registration trial where we co-administered gadolinium with the drug, the label would say drug plus gadolinium. It could not just be drug, okay? And so that's true if you're doing any 2 drugs. It's whatever you're doing at the registration trial, that becomes the label. So you can do it. If you think that using 2 drugs together is the way to approach it, then you can do that. It's just if the 2 drugs are in the same company, that's easy. If it's in different companies, then it's some kind of cooperative agreement between them, and that's where the lawyers get involved, and I just wait to hear something back from them.
And Dr. Turkmani asks, can you comment on the current status or what your thoughts are on selective endovascular drug therapy for brain tumors and glioblastoma?
Yeah, so that falls along the lines of the blood-brain barrier disruption experience. I think that there have been a lot of interesting trials there. There have been some successes for gliomas, not so much. One of the the idea of doing selective drug delivery, I still think is an interesting idea. It's one where we really do need to have a lot more in the way of evaluation of the tumor tissue afterwards to see what's really happening. Right now, it's all been clinical endpoints, and there's this black box, and we don't really know what's going on. And if it was really working, we'd all know about it. So we need to understand whether or not we're really getting drug there and what it's doing. A lot of it is based upon diffusion. You have in these tumors, you have expanded interstitial space. You have tissue pressure that can often push the drug out. We really don't know what's going on there. So I think with Edge's approach with the microdialysis catheters, perfect. That's a great way to start asking those kinds of questions. In fact, then you get time data, which we don't get from a single sample. And I think if I were to do a development program with intraarterial drug delivery, I'd want to have a microdialysis catheter. If we had something we could effectively measure, I'd want to do that.
Edge, I think it's okay if you steal that idea now.
Just as
I have a
long
list.
as
I have a
we're
list from this talk
collaborating
alone.
institution, okay, that's all I ask.
Very good.
Great. Well, thank you again so much. I think if we don't have any more questions, anybody else have any questions? I think we'll wrap it up there. But Dr. Vogelbaum, thank you so much. I think that the next generation of clinical trials needs to follow this talk and the guidelines and the advice that you have, and we need to change the ways that we're doing things. And so thank you for taking the time to tell us about your thoughts and all your experience and successes. And hopefully the younger generation could follow in their footsteps and take it to the next level.
Well, great. Do it better than we did.
Really fantastic. Thanks very much.
Great talk. Thanks all.
Good to see
Thanks
you.
so much.
Thanks.